Hello students, welcome back to my channel. So today we are going to study about the life cycle of Rhizopus. Rhizopus is a member of Zygomycetes, and Zygomycetes is a division of the Phycomycetes. So we are studying one example of Phycomycetes, you can say, that is Rhizopus in detail. First of all, you recall this, that Zygomycetes members are having cenocytic hypha. If you remember, they are having cenocytic hypha. Cenocytic hypha means they are having multinucleate hypha. So here we will find there are two types of hyphas are there. One is just parallel to the substrate, another is penetrating inside the substrate. So this is cenocytic hypha. Okay, so this is multinucleate condition. And this is the hypha which is going penetrating inside it. So this part is known as stolonifer hypha, which is behaving like a stem, because we know very well that there is subaerial stem modification known as stolon, and this is working like a root. So this part is known as rhizodial hypha. So these two hypha are vegetative. One is behaving like a stem, one is behaving like a root. So this is root-like, and this is behaving like a stem-like. Now on top, you are finding it is having asexual hypha also. So that because we know very well that they reproduce by making the aplanospores in the sporangia. So this is your sporangiophore, and the tip of the sporangiophore will be swollen to bear the sporangia. So this is the sporangia, and this is the stalk which is known as sporangiophore. So this nuclei I am using with the white color. You can understand, please. This is the white nucleus which is entering inside it also. And as a result, the nucleus is more clustered towards the periphery. More clustering of the nucleus is occurring on the periphery, and center, nothing is there. So this central part is known as columella. And this peripheral part which is having cenocytic condition, this peripheral part is known as sporangioplasmaria. Okay. Now see what we will observe after some time. Let me draw once again one stolonifer hypha, this one sporangiophore, so that you can understand easily. This is your, I'm drawing it second diagram just to make you learn how the things are going. And inside this you are finding the aplanospores are found. These are your aplanospores. Means the cytoplasm is laid around nucleus to make the aplanospore. Are you getting it? These are the way how the aplanospore is formed, leaving the center area which is known as the sterile area, columella. The center, this part will be known as columella, which is a sterile, and at the periphery you are finding the formation of the aplanospore, also known as sporangiospore. So this is the way how they make their asexual spores. Another what you have to remember, they at one point there you will be finding many sporangiophores will be there, which is bearing at the tip, they are bearing sporangia. From one point many will be coming. This is the difference with respect to the mucor. In mucor only one will be coming at a time. Okay, because two are the examples of the Zygomycetes, that is Rhizopus and mucor. So this is the condition present because from one point many are coming this way. That is why Rhizopus is also known as pin mold, because they give an appearance of a pin like. That is why it is known as pin mold. Now see, at the time of sexual reproduction, let us assume this is your one hypha. Okay, this is the plus hypha and this is the minus hypha. Means heterothallic is observed, and heterothallis was first observed in Rhizopus stolonifer. Means two different thalli are there for the sexual reproduction. The heterothallis was first observed by A.F. Blaxley. A.F. Blaxley, and he observed it in the Rhizopus stolonifer. This is again a PMT question, please understand it. Rhizopus stolonifer, it means, first of all, what does it mean? Heterothallis means two different thalli will be there. One is working as plus, one is working as minus. Now, thalli of different strain will release chemical trisporic acid. Okay, so what will be the response? If the chemical is released by the minus strain, then the plus strain will make this swelling. Okay, so this swelling is known as progametangia. Same way, the minus strain will also make up progametangia in response to the chemical which is released by the plus strain. So this is the formation. First step is the formation of the progametangia. Clear? Next, what will happen? Slowly, slowly this gametangia will grow. Now this gametangia will become into big in size. With multinucleate condition only, so we will say cenogametangia. Ceno word means multinucleate. So we call it as cenogametangia. Same with respect to the minus hypha also. The minus hypha will also give rise to cenogametangia. So the cenogametangia formation is done. After that, the two hypha will come near while the cenogametangia formation will be going on, and cenogametangia will become into the suspensor, and this structure which is now developing into cenogametes. Cenogametangia will have cenogametes. Same, this will also come near. The cenogametangia will have cenogametes. So this is the cenogamete formation. This structure is cenogametes. Now this hypha, this stalk, is going to bear the zygote. We will say this is zygophore. Same here, this is known as zygophore. This one, understand the diagram. Okay, this is known as zygophore, this part. After this, what we will observe, that the two cenogametangia with their cenogametes will fuse. This side cenogamete and this cenogamete, they are going for fusion. So they are making this structure fusion is there. This structure with a thick wall now. This is known as your zygospore. Okay, this will be zygospore, And see, the wall will be between them is dissolved, and the nucleus of plus are there. These are the nucleus of plus, let us assume, and these are the nucleus of the minus. They are now will be fusing it. Okay, after fusion, they will be making the zygospore. Now, what will be the zygospore? How the zygospore will germinate? So they have made this structure zygospore where the diploid condition is present. I'm using this color for the diploid conditions where the fusion has been done. Many nucleus will be there. Okay, so these are the fused conditions of the inside the zygospore. This is zygospore. And this will germinate to form, zygospore will germinate to form one again germ sporangia. It will germinate to form again one germ sporangia. So this is germ sporangia. And this is the germ sporangia 4. And inside the germ sporangia, the meiosis will occur. This is the site where meiosis will occur. Now let us assume here, suppose there were 10 diploid nucleus were there. Okay, 10 diploid nucleus were there. So only one diploid nucleus will go for meiosis. Will do meiosis. So after doing the meiosis, it will produce 4 types of spores. Okay, and only one type of spore will be remaining 3 will finished off. Only one functional spore will be left. Which will develop into the mycelium. Again, they start the cycle. So I hope this concept which I explained you, this will help you to understand the topic easily. Your Rhizopus, which is an example of the zygomycetes. Thanks for watching, class. In the next video, we'll be discussing about the life cycle of Puccinia because few questions they ask from the Puccinia life cycle. This video is especially made for the NEET preparation. I hope this video will help you to understand the life cycle of Rhizopus.